Hello, everybody. Thank you for joining us today. Hopefully you can hear me all right. I'm, it's been a little while since we've had a Roy Ball guest speaker come present to us, but I'm really excited about who we have coming, who's here with us today. So I'm going to do a quick introduction for Sahib Khalsa. He's an MD, PhD trained associate professor and director of anxiety disorders research in the Department of Psychiatry and Biobehavioral Sciences at UCLA. He also still has his foot in the clinical world, taking care of patients, a supervisor in the UCLA Anxiety Disorders Clinic and the UCLA Veterans Affairs Anxiety Disorders Clinic. So he's also based in the VA, where he teaches psychiatrist, being trained in the clinical management of anxiety disorders and co-occurring mental health disorders. He's also a particular interest to our Roy Ball Center. He's the founding director of the UCLA Healthy Hearts Behavioral Medicine Program, which is an interdisciplinary endeavor started with the UCLA Cardiac Arrhythmia Center, focused on treating anxiety and stress-related disorders in individuals with cardiac arrhythmias and other medical conditions. So a lot of shared interests with different studies that have been conducted through our Roy Ball Center. I mentioned he's a board-certified psychiatrist and physician scientist who completed training in medicine and neuroscience at the University of Iowa Medical Scientist Training Program and did subsequent training in psychiatry at the UCLA Neuropsychiatric Institute. And he leads an active program of research on the neurobiology of interoception and mental health funded by NIMH. And today he's going to tell us a little bit about his research with a focus on interoception, which again has been one of the major interests that we've had in terms of developing interventions through our Roy Ball Center. And Sahib asks people to save their enter questions in the chat as you go if you'd like, but we're going to save the discussion section for the end of the talk. And we'll have a little bit of time to go a little bit past 1 p.m. for those of you who can stay late. Okay, Sahib, on to you.
Okay, can everybody hear me okay?
Yes, you sound great.
Okay, great. All right. So first of all, thank you for having me. The title of my talk is Interoception and Fear, New Insights into Brain-Body Communication. And while you're looking at my disclosures, which I have no disclosures related to this talk, you may be wondering why I have a bandage on my face. This is your friendly public service announcement that sun exposure is cumulative. So please do wear your sunblock. Okay, so I'd like to start out just by acknowledging all the lab members, current and former, and collaborators who have really supported me in generating the work that I'm about to show you, as well as funding agencies, both public and private. I'd especially like to thank Ian for inviting me to be here, and all of you at the Columbia Roy Ball Center for listening in, and Darlene for coordination. So I'm going to start with an overview of interoception, then I'm going to talk about some new insights into interoceptive pathophysiology of psychiatric disorders, both in the medical sphere, in the psychiatric sphere, as well as the medical sphere. And then I'll talk a little bit about a new treatment-related development that I'm particularly excited about. This image that you see is actually on the right is the image of the page views on Wikipedia for the term interoception. So one thing you'll probably immediately notice is that in 2017, there's a big jump. That's because I had a couple summer undergraduate students sort of go to town and revamp and try to make a little bit more accurate the page view content. But you can see that even since then, sort of public interest in this concept has been growing as evidenced by the page view increases. Now that's mirrored by the publications in the English language, which you can see here. One thing that's clear is that interoception is not a new term. It goes back to 1906 when Charles Sherrington defined it. But you can see prominent examples easily going into Pavlov's work, William James, Claude Bernard, as well as Darwin. One thing that is also clear from this graph is that interest has sort of fluctuated in the preceding century, but really has taken off quite a bit. In particular, when I moved to LIBOR, I was fortunate to be able to organize the first summit on interoception and mental health. A couple of years later, NIH caught up and organized a neuroscience blueprint interoception. They now have one of these every year. In 2021, the Nobel Prize was awarded in part for the discovery of the piezo mechanoreceptor, which Artem Patapoutian, during his address, sort of characterized as a interoceptor in the Sherringtonian sense. So interest is growing. Earlier this year, the NIDDK Institute had a workshop on interoception and gut feelings. So it's certainly a space to watch. That said, this is an area, interoceptive neuroscience, as a field, is in its infancy in many ways. This is not my kid, by the way. I just got this from the internet. But what it means as you're listening is that there's many gaps as well as many opportunities. There's incredible potential for growth, but there's many ways that progress could be prematurely stunted. So this is just a visual reminder of the importance of keeping this early developmental stage in mind. Okay, so how can we sort of define or think about interoception? One of my goals for today is that you would be sort of become a mini interoception expert, and you can sort of express that to other people who ask you about this kind of strange term. So at this meeting that we organized, it culminated in this special issue. We came up with a consensus sort of agreement of kind of how can we sort of define it at least at a one point in time. What we came up with was that interoception is a process by which the nervous system senses, interprets, and integrates signals originating from within the body, providing a moment-by-moment mapping of the body's internal landscape across conscious and unconscious levels. So there's a lot packed into that, right? It's a process, nervous system, so it's not just the brain or the central nervous system, right? Brain and spinal cord. You've got sensing, interpreting, integrating. The signals are from within the body, and it's a moment-by-moment mapping across conscious and unconscious levels. Now, if you're a human researcher, you're probably more familiar with another term. Some people have described it as sort of the second definition, or interoceptive awareness, which we defined as the conscious sensation, interpretation, and integration of information about the state of inner body systems. So the conscious piece is sort of the key. So if we were to pictorialize this, if interoception is an iceberg, then you can think of sort of interoception as the small piece of what's above the surface. And what we attempted to do subsequently is to provide additional definitions in relation to the features or components of interoceptive awareness. So there's an attentional piece, there's detection, there are magnitude estimations, there's discrimination or localization. Accuracy or interoceptive accuracy is a particularly commonly studied phenomenon. And oftentimes you'll see in papers, people equate interoceptive accuracy with interoceptive awareness. But the point I'd like to make is that interoceptive awareness encompasses all of these different components. Insight is sort of a metacognitive evaluation, sort of how accuracy corresponds with confidence. And it's certainly something that's clinically relevant. There's a host of self-report scales that are associated with measurement of interoception. And then there's, of course, sensibility or the trait. So there's a lot there. Methodologically, I've been focused on trying to help operationalize how can we measure this. So here's a very clear example. I'd like you to just sort of follow along with me. If I asked you to just simply for a couple moments pay attention to your heartbeat. Okay, so wherever you feel that sensation in your body, find it and focus your attention on it. And if you've got it, great. If not, don't worry. Now I want you to start counting every time you feel a heartbeat. Or tap a button, or tap your thigh or the table every time you feel it. You can imagine that I'm playing a tone every time your heart might be beating. And then finally imagine that your heart begins to race, right? So each of these are different tasks that are available in the literature that measure different components of interoceptive awareness in the cardiac domain. Okay, the challenge is that there are many domains beyond simply the cardiovascular, and they all can be measured according to this way. And of course, in real time, the signals that are being relayed from the periphery to the central nervous system vary in frequency and amplitude quite a bit. And your nervous system is monitoring these things all the time. So there's quite a bit of information there. Now, I'm going to talk with you today a little bit about examples in the cardiac and respiratory domain, as well as if there's time in the gastrointestinal domain. But first, I want to provide a little bit more definition in terms of the physiologic processes that are often ascribed to interoception. So we can kind of divide these grossly into non-painful or painful. And one of the things you'll immediately be saying is, well, whoa, there's kind of a lot there. So that's no doubt. Another thing is, and if I were in a room with you, I'd ask people to raise their hands. If you feel comfortable digitally raising your hand, you can. Anybody ever have a kidney stone? So this is a X-ray and an electron micrograph image of a kidney stone. So some of the most intense pain people will imagine. I see at least one person raising your hand. Thank you, for the person who did that. So 10 out of 10 pain. The last talk I gave, somebody said it was 12 out of 10 pain, right? Imagine one of these passing through the ureter, as is shown, sort of grating its way along the epithelium. Incredibly painful, right? Another kind of key distinction beyond the painful, non-painful is that interoceptive signals usually occur outside the realm of conscious awareness. And then they also occur across a spectrum of arousal. So a couple of years ago, I was fortunate enough to go hiking in a blizzard. I was giving a talk in Utah, and it was quite a lot of fun. Snowshoeing up in the mountains, that was a happy side, right? You can see quite a bit of a workout, heart racing. But I treat patients routinely who the very prospect of increasing their heart rate at all induces quite a bit of anxiety and fear, and they avoid it. In fact, pathological avoidance of sympathetic arousal is kind of quite a hallmark. So there's sort of a discrepancy there because putatively this is the same level of arousal, but you can have a very positively or negatively balanced response. The other aspect is that from a systems perspective, we often talk about a brain-body feedback loop. So you have organ systems that are communicating with nervous systems, and then you have nervous systems communicating with organ systems. And there's a ton of these systems, as you can see on the left. And there's multiple components of the nervous system, right? Central, peripheral, autonomic, somatic, and enteric. And so part of the notion of this loop is that sometimes signals that go from an organ system to a nervous system elicit so the sort of afferent input elicits an efferent or regulatory input that can then potentially change the sensation or the signal. So you have this sort of feedback loop, and it becomes often very difficult to tease apart the afferent from the efferent, the sensory from the regulatory aspect. Also, I haven't even described or talked much about the microbiome, which is a whole nother area of science. You can sort of think of a world as a world within a world. So pretty complicated. From a neural circuit perspective, what I like to define an interoceptive neural circuit is as a network of nerve cells that are connected through sensors, and there are various types of those, through into neural pathways that create networks that we can group on the basis of function. But the definition of an interoceptive circuit is really made in the context of a function, and it's a function that is self-related. It's not about looking at other living or non-living creatures. It's inherently self-related. We're learning a lot about the transducers of neural circuits, and I won't go into all of these in detail. This paper does a reasonable job of characterizing some of the different transducers or receptors. And we know that there's we know a lot about the sort of peripheral neural inputs that are vagal, glossopharyngeal, spinal, that go to the brainstem. But it turns out, and there's a lot of animal literature that's actually growing and enhancing our knowledge of this peripheral circuitry. But when it comes to the central nervous system, we know a lot about an increasing amount about different brain areas. It's not just the insular cortex. And I'll illustrate that with some data. But there's kind of a disconnect such that we know a lot more about the peripheral areas leading into the brainstem in animals than we do about the central areas. And we know a lot more in humans about the central areas than we know about the peripheral relays. There's probably other areas beyond what I've shown here that are involved in interoception. And as an example, this was a paper published earlier in the year that suggested that there are mitral cell neurons in the olfactory bulb that actually relay heartbeat signals within a 20 millisecond delay of the pulsation. So that's faster than baroreceptors relay information from the peripheral arteries into the brain, which is at about a 60 millisecond delay. So why is that? What's the relevance evolutionarily of having such a rapid access to a pulsation? Another this is like 2 weeks ago, this paper was interesting, just in terms of the sensory integration side of things, provided evidence that humans with congenital anosmia actually seem to breathe differently. So how is it that the inability to smell is creating sort of a different pattern of breathing function? That's kind of a curious phenomenon. So I want to shift gears a little bit to the clinical realm, and in particular, this notion of symptoms. A lot of what we see in patient care is patients come to us when they perceive symptoms. Now, the perceptual processing of interoceptive symptoms is tightly regulated, but once there's a sort of a homeostatic deviation, let's say a patient comes to you in the ER, or they come to you in clinic complaining of palpitations, dyspnea, and flushing, you may have a differential diagnosis immediately that spans medical and psychiatric sequelae. Same thing for hunger, abdominal fullness, or bloating and cramping. So we often think that many of these symptoms may reflect an organ-specific pathology, but it may also be possible that some of these symptoms are driven in quite an unconscious manner that relates to an abnormality of homeostatic functioning called allostasis. So I like to define homeostasis and allostasis as the following. Homeostasis is sort of a reactive regulation. You imagine that this curve here reflects something like, let's say, blood pressure or blood glucose levels. And you have a drop in your blood glucose levels that triggers a homeostatic control. Let's say you have a hypoglycemic event and then you eat. But what if you know that every time your blood sugar drops, you start to get a little bit irritable and lightheaded? So you could enact what's called allostatic control, and maybe you eat a candy bar beforehand so that the next time your blood sugar drops, your homeostatic control is buffered and you don't end up into a dangerously low hypoglycemic state. And then when that is no longer required, you have allostatic control resetting. So this is sort of a physiologic example, but from a disease example, this was allostatic load or overload is something that Bruce McEwen highlighted as a very key element in the response, the pathological sort of response from an adaptive to a maladaptive state across many different diseases. So the idea is that when this predictive regulation really can't keep up with the body's needs. From a perceptual standpoint, we think of symptoms as being at the top of a hierarchy, as an explicit sort of hierarchy, but we have sort of two layers of processing. We have explicit and implicit, and we even have implicit metacognition in addition to explicit metacognition. So one of the questions is how much of what people are reporting actually comes from this domain as opposed to this domain. So are interoceptive symptoms really driven from an actual body change, or are they driven by a neural circuit change that doesn't involve the body, or perhaps both? I'd like to point you to this textbook, which recently came out. It is a textbook on interoception, and there are sort of three conditions that I would like to specifically point your attention to, although all of these in the symptom column reflect symptoms that relate to the diagnostic characteristics of various psychiatric disorders that you see listed. So these are diagnostic symptoms. In psychiatry, we don't have any biomarkers. Well, we have one biomarker. In the Q&A, I'm happy to talk about that. But really what we do is we diagnose based on collections of symptoms with the assumption that there's an underlying sign and pathophysiology that's connected to it. But thus far, we don't really have any biomarkers. So this symptomatic description really doesn't provide any information about underlying mechanisms or causes of these disorders. One hypothesis that I'd like to argue is that by linking brain and body, interoception may provide one component process for arriving at a better understanding of certain mechanisms and causes underlying disturbed mental health. And that brings me to sort of one take-home message from a scientific standpoint, and that is that perturbing peripheral organ system signals is an optimal way to understand how interoceptive neurocircuitry causes symptom perception to occur in a disorder-relevant fashion. So this is really going to set up some of the work that I'm going to show you from my lab. And we'll move on to the second part of the talk. I'm going to focus first on heart-brain communication. And first, again, if I was in a room, I'd ask for a show of hands. You can raise them virtually if you'd like. But I'd like you to sort of think back to that example where you were paying attention to your heartbeat, certainly if you're doing it now. And, you know, just yes or no, could you feel your heartbeat right now without taking your pulse? Turns out that, okay, I see some thumbs up. I don't know how many that is. It's too bad we don't have a poll. Oftentimes, when I give this talk, usually the answer is somewhere between half to 2/3 of the people felt like they could feel their heartbeat. But it turns out under sort of standard laboratory settings, when you use sort of a visceral psychophysical, like a signal detection approach, where you can tell at an individual level, only about 1/3, so 1 out of 3 people, are actually able to accurately perceive their heartbeat signal. This is true, by the way, whether you are a long-term meditator. This was one of my first empirical studies. It's even true if you have somebody else's heart beating in your chest. So this was a heart transplant study that was recently replicated as well. We know a lot about brain areas that are active when you're attending to your heartbeat sensation. So here, this was an early study showing that there was a network involving the insular cortex and somatosensory and anterior cingulate. But this correlation, while it looks very strong, has to be couched in the context of the fact that most people actually don't feel their heartbeat when they're sitting there under resting circumstances. So how do you explain this sort of discrepancy between being confident or fairly confident that you felt your heartbeat, but not really being able to show that? Well, I'm going to try to illustrate this sort of conundrum with a couple images. So this is sort of an example of how sensory expectancies can shape perception. So I'm going to start with a few exteroceptive examples here. Does anybody see the face? How about now? Now, if you look at this, you probably can't unsee it, right? I'll give you another visual example. Can anybody make any sense out of this signal? Looks kind of like just a bunch of odd shapes to me. But how about now? Are you able to see the letter B's? Probably, right? So one explanation for these kinds of effects is that the brain sort of fills in the gaps, particularly with ambiguous stimuli, as a way of making sense of the external environment. So I want you to, we're going to try to sort of extrapolate that into the interoceptive realm. We're going to do that by using this sort of example, where imagine you have a, actually, I want to start over, because I want to start by shifting to fear for a second. I had fear in the title, and I want us to start thinking about interoceptive signals, and particularly ones that might be fear-inducing. But in order to do that, and to illustrate this sort of divide, we need to kind of also have the conversation about what's anxiety and fear. It turns out that the experts are quite divided. I'm just going to illustrate that here. So these are sort of 4 experts who describe it in various ways. Joe Ledoux, fear is a conscious awareness that you're in harm's way. So conscious awareness. Kay Tai, who's done a lot of animal research, fear is an intensely negative internal state. Mike Fanzelo, who's here at UCLA, fear is a neural behavior system that evolved to protect animals against environmental threats. So here you have fear being associated with harm, negative internal states, and threat. And then here's an interesting one from Lisa Feldman Barrett. It's fear is constructed in an animal's brain as a plan for assembling motor actions and visceromotor actions that support them, as well as the expected sensory consequences. So quite a bit of information there. You may have your own opinions and will have to sort of make your own decision. That's for fear. But what about anxiety? I see a lot of patients with anxiety disorders. The way that I like to sort of define anxiety in relation to fear is I think of fear as more of a threat response when there's actual perceived tissue danger, whereas anxiety is a threat response in the absence of a threat-inducing situation. So there's no actual or potential tissue danger, but the individual perceives that there's a threat response. Okay. So in terms of how we can have this sort of divide between a sensory signal and an apprehension that creates an abnormal perception in the body, we can think of sort of a top-down versus bottom-up interoceptive dysfunction. And we can think of people as having potentially different phenotypes. So here's an example of an intact prediction signal of a prospective aversive bodily state. This I took from a great paper by Martin Paulus and Murray Steen. It's rather old now. But they were illustrating how you could have this sort of top-down dysfunction where you could have an exaggerated prediction signal of a prospective aversive bodily state. So the prediction is so abnormal that it creates a sense that there is an abnormal state in the body. And what they sort of differentiated this from was from a bottom-up dysfunction. So you could have a bottom-up dysfunction, which is this low fidelity signal in red, where you can see that periodically it rises above the threshold of noise. So you can see that it's hard to differentiate this signal, which seems quite pertinent, from this noise, whereas this individual in black here really only has 1 or 2 points where there's a high fidelity signal to noise change that would be worth paying attention to. So you can imagine somebody with this sort of top-down dysfunction or a combination of a top-down and bottom-up dysfunction perceiving signal when there's noise, as well as perceiving signal when there's signal. And so how do you differentiate that? Computational modeling is one proposed solution. And I'm not going to go into this in detail. You can read, these are 2 excellent papers that provide an overview. But if you think back in relation to the constructed definition of fear that Lisa Feldman Barrett gave, the general idea is that you have sensory signals coming in that meet with priors or prior experiences of those types of signals that are informed by an internal model. And that perception is the combination of the sensory signals coming in with your priors. Priors are operationalized as predictions. And so when you have a difference between your prior and the sensory signal, you have a prediction error. And when you repeatedly have prediction errors, that can inform your model of the body or of the world. It's informed by metacognition and forecasting. But you can see that in combination, what you do in relation to what you're feeling and predicting is going to impact how you act, which can then keep things going in a different, move things in a different direction or keep them the same. So you can use computational models to actually operationalize each of these components numerically. You can study it in non-human animals as well. And so I think that's sort of one area where there's been a lot of activity. I want to speak a little bit to disorders of fear that we often see in the clinic. This was a paper where we attempted to really elucidate a potential role for interoception in relation to fear learning and PTSD. And the point that we were trying to make with this image was that there's sort of a susceptibility window. PTSD is a condition that occurs, it requires an event in order to occur. It generally doesn't happen below the age of 1, and it generally doesn't happen in a very advanced age. So you could argue this is a pretty wide susceptibility window, but there is a susceptibility window nonetheless. And it occurs in the background of traits that people bring to the traumatic event. So there's a pre-morbid sort of brain-body feedback loop. You can think of this sort of prediction error processing that I showed you earlier. And that sort of bottom-up or top-down dysfunction. Maybe some people are more higher or lower on that spectrum. But then when you have a traumatic event, there's a huge surge of afferent input from the body into the brain. There's fear learning that happens. And there's also safety learning. And we're learning a lot about the neural architecture of safety learning and how it has some overlap with fear learning, but also some distinction. Now, some people may have abnormal fear learning and also abnormal safety learning, and they develop PTSD, sort of characterized by this cartoon, whereas others, when they have optimal safety learning, they don't develop PTSD. We know that most people who experience a traumatic event, in fact, don't develop PTSD. And then, of course, the goal of treatment is to normalize this abnormal feedback loop. So what are some potential fear learning mechanisms that relate to interoception that might play a role? We sort of proposed in relation to some other studies that are out there that if you use sort of the classic conditioning model of conditioned fear, this is just an example where there's a sexual assault where an individual has an unconditioned response that is conditioned by perpetrator's cologne. So next time let's say they smell somebody wearing that cologne, they have this unconditioned response characterized by increased heart rate, heart palpitations that they perceive, shallow breathing, and dyspnea that they feel, sort of choking sensation, difficulty breathing. Well, what we added to this was this notion that you might have another layer of learning, what we called higher order conditioned fear, where you have a second conditioned learning stimulus, where now you actually become conditioned to subtle changes in heart rate and breathing rate, or the perception of subtle changes in those that then lead to an unconditioned response. Such that you could be conditioned to other environments as well. So your CS2 could be conditioned to other CSs, for example, the treadmill, such that in a safety context like the gym, you know that exercise is good for you. It's an anxiolytic. You're trying to work out, and yet the CS2 triggers an unconditioned response. That could lead to negative affect, negative cognitions, and then avoidance of these conditions and maintenance of PTSD. So this is sort of one proposed model. We know a lot about sort of brain regions that are relevant for fear learning and PTSD. This is just a schematic cartoon. And in the paper, we sort of argued that in order to better understand the role of interoception in fear learning and PTSD, you need to keep some of these other contexts in mind. In the exteroceptive context, the interoceptive context of the individual, their pre-morbid traits, whether they have a high or low sensitivity, what sort of allostatic set point they might have been in at the context of the trauma. Were they already under a high allostatic load? And then what's their predictive processing phenotype? Are they more likely to have a high precision weighting of prediction errors or low in other weighting? In other words, are they likely to make a lot of meaning or not a lot of meaning out of these signals? This is, of course, relevant to a variety of different contexts. This was a brief view that Negar Fani and I wrote in relation to racial discrimination, sort of a hypothesis that initial exposure to racial discrimination could trigger heightened levels of sympathetic arousal and cardiovascular stress responses in the body, negative emotional states, and threat memory encoding via threat circuitry. And that repeated exposure to racial discrimination could elicit the same type of cascade, leading from a shift from reactive homeostatic to predictive allostatic responses, which could then elicit emotional numbing, disrupt interoceptive awareness, and promote dissociation or separation from the body or detachment as sort of a protective response. And of course, with something as complex as racial discrimination, it's clear that there are a variety of other contextual factors that are social, hormonal, environmental, and structural. Okay, so that's a lot of theory about interoception. You're probably interested in hearing about some data. So one way that I've tried to sort of make inroads in understanding about interoception is to use what I sort of loosely call a visceropsychophysical approach. And that's leveraging what we know about the hypothalamic pituitary adrenal axis, so that sort of the stress axis, if you will, but not looking at cortisol, but rather epinephrine and norepinephrine. So adrenaline, which is a acutely acting fight or flight neurotransmitter. And we know that that's activated during stress and during trauma. What I've utilized is not adrenaline, but a drug called isoproterenol, which is a beta adrenergic agonist. You can see that's very similar to epinephrine. The advantage of isoproterenol is that it does not cross the blood-brain barrier. It stimulates beta adrenergic receptors in the heart and lungs, causing increased heart rate, heart contractility, bronchodilation, and feelings of palpitation and dyspnea. When you induce it, inject it intravenously, this is just an average curve in 21 healthy individuals using a randomized double-blinded placebo-controlled with saline. You can see that you can get a nice range of homeostatic perturbations. The normal heart rate ranges from 60 to 100 beats a minute. So you can see a 0.5 microgram dose, you get a little bit of a bump. With a 2 microgram dose, you get a larger bump. But in general, after about 4 minutes, people's heart rates stabilize and come back to baseline. You can see that there's a peak in the response to the heart rate, and then a recovery that's early and then late. We're interested in perception. So what we paired this with is with a dial, and the purple is the dial. This is a 2 microgram dose on average in healthy individuals, and you can see that in double-blinded placebo-controlled conditions, the heart rate response is followed by a perceptual response, which tracks quite nicely the change. There's a little bit of a delay, but you can see that on average, you're getting up to a heart rate of over 100 and a intensity rating of about a 6 out of 10. We've done this using a variety of different doses, and I'm just going to show you these are overlap dial curves. Pay attention to the 2 microgram and the 0.5. So you can see that you get a larger heart rate response at 2 than you do with 0.5. You still get a bit of a heart rate response with 0.5, but you get more with 2, and you get a full detection difference. So everybody under blinded conditions feels their heart rate change with 2, whereas with 0.5, it's somewhere in the middle between the placebo and saline and the 0.5. We've done a lot of studies using this particular perturbation. I'm not going to go through all of them. I'm going to highlight a couple. We've adapted this approach to the scanner. So here's a nurse sitting in the scanner room. The participants here with an IV in place. Well, this is what the participant sees. They're using sort of a speedometer like display to use the dial to rate their real-time rating of cardiac and respiratory sensation. We do cardiac and respiratory because they feel both approximately at the same time. And then we get retrospective heartbeat and respiratory intensity ratings. Isoproterenol has a short half-life, so you can do it repeatedly. And this was a study where we looked at whether there were differences in the brain and body in individuals with generalized anxiety disorder. The first thing I want to point you to is that we didn't see group differences in heart rate response at saline or at the 2 microgram dose. But curiously, at the 0.5 microgram dose, that sort of sub-threshold, we saw a higher heart rate response in the GAD individuals. And that was also matched, again, by a higher heart rate response, a higher dial rating response, indicating that the increased heart rate change elicited increased dial perception. I like to think of this as sort of a potentially left-shifted dose-response curve, although we don't really have enough doses in between in this study to really get a proper curve. Our hypothesis was that individuals with GAD would have heightened responses in the insular cortex. What we saw was, while we saw a dose-related response in the insula, no difference between the patients, but instead a hypoactivation of the ventromedial prefrontal cortex that, again, only at the 0.5 microgram dose. So the way we sort of, and this was actually strongly correlated with their retrospective rating of heartbeat sensation. So the way we interpreted this was that there may be a form of autonomic hyperreactivity, sort of the increased heart rate response in the GAD individuals was associated with an increased peripheral response, but also that the increase in hypoactivity in the GAD patients in the VMPFC was evidence of a failure of the VMPFC to constrain low to moderate levels of sympathetic arousal. So the idea being that sort of the VMPFC is trying to put the brake on sympathetic arousal, but is really failing. And that in combination, you have this sort of exaggerated response. So we've done some subsequent analyses to try to look at the relationship between the VMPFC and the insula and other areas of the brain, like the amygdala, that we know are associated with anxiety. We did a functional connectivity study, and we found, using 2 different analysis approaches, a frequentist standard FC response, FC analysis, and a Bayesian one, that the ventromedial prefrontal cortex and the posterior mid-insula, the area that was encoding the ISO-induced sensations, were the most abnormal areas in GAD. And in fact, that connectivity in this area was associated significantly with something called anxiety sensitivity. So anxiety sensitivity you can think of as a trait that relates to the fear of experiencing fear-inducing stimuli. So there's something going on in the VMPFC and the insula. This is certainly related, matches up with sort of more recent theorizing about fear. So here's Joe Ledoux talking about fear not just involving the amygdala, but also sort of involving what he calls higher order circuits. This is sort of Dean Mobb's review looking at fear and safety learning. And what you can see is that you've got the ventromedial prefrontal cortex and the insula involved in fear and safety learning. And the safety learning sort of involves this sort of anterior portion of the VMPFC, whereas the fear learning involves this more posterior portion. There's a really nice convergence with animal literature. So this was a study that I sort of very loosely, this is from Carl Deisseroth's lab. This is an animal study using optogenetics that I kind of like to describe as the mouse equivalent of the isoproterenol experiment. Basically, they used a virus to induce opsins in the heart of these mice, such that when they shone a light using a telemetry vest that these little mice wore, they could increase heart rates above the normal resting heart rate of a mouse, which is 600 beats a minute up to 1,000. And then they could image using tissue clearing which neurons were most sensitive to the heart rate increase under anxiogenic learning contexts. And they found that it was two areas in particular, the posterior insula and the medial prefrontal cortex, which are direct analogs of the areas I showed you in GAD. They found that if they inactivated the insular cortex, they could abolish the sort of fear phenotype, whereas they couldn't show that with the medial prefrontal cortex. So there are some open questions here about why silencing the insula but not medial prefrontal has an effect in mice. And then why is the VMPFC but not the insula so relevant in humans? And there's another question that I have about whether there are other neural indicators of cardiac interoception in GAD that we might be able to look at. We've begun looking at some of these in relation to something called the heartbeat evoked potential. So this was a follow-up study where we found that individuals with GAD had a increased heartbeat evoked potential, again, only at the 0.5 microgram dose, and that these amplitudes were associated, again, with anxiety sensitivity. So there seems to be something related to a fear of fear, and perhaps that is involving a VMPFC to insula circuit. From a therapeutic standpoint, how could we potentially intervene? I'm intrigued by an area of the brain stimulation world called focused ultrasound that people are starting to study. This is a recent paper. This is just in healthy individuals that a colleague, Win Ligon, showed in healthy individuals using this sort of randomized sham-controlled approach that by delivering focused ultrasound, which basically the unique thing about ultrasound as opposed to TMS is that you can provide very focal and deep stimulation that's inhibitory to regions of the brain that are very difficult to reach, that TMS cannot reach. So he showed that when delivering focused ultrasound to the posterior insula, they could attenuate the amplitude of the heartbeat evoked potential, and that it had some influence on symptoms. So I believe that there are some promising approaches ahead, both in terms of our understanding of the pathophysiology of fear-related cardiac processing, and there may be some relevance in terms of this particular type of approach that's minimally invasive that can sort of focally target deep limbic structures. Now, I only have a few minutes left before questions, so I want to turn to this therapeutic approach that I'm particularly intrigued by. I'm going to start with some quick case examples. Some of you in the audience are likely familiar with, or maybe have even treated patients with these conditions. So this was a patient who I treated at UCLA. He was a 66-year-old man who had an idiopathic ventricular tachycardia, no history of psychiatric conditions, and had received placement of an ICD to prevent VT. Now, after he got his first shock, just a single shock, he had fear conditioning. So he wouldn't leave a 5-mile radius from his home, which was near the hospital, for an entire year, because he was worried that if he left, he would get shocked and die, and wouldn't be able to be resuscitated at the hospital. He wouldn't shower alone for 3 months and was afraid to be alone by himself in a room for more than a year. There's another individual treated at UCLA, a 35-year-old man who began spending all his time inside his local hospital after receiving recurrent ICD shocks. His explanation was that this was the safest place to be in the event of another shock. He remained in the hospital and public areas for months with significant anxiety upon leaving. He even purchased a stethoscope to better listen to his heartbeats and claimed he could accurately predict his arrhythmia within a latency of about 3 seconds, although he wasn't able to do anything. These are sort of cardiac examples. There's sort of a GI example in relation to anorexia nervosa. So this was a woman with AN who sort of felt full and bloated after eating several spoonfuls of cereal, whereas she likes feeling empty and light. And this feeling fluctuated with relapses, but never really completely resolved. So how could we sort of these are complex conditions that don't respond very well to treatment. How can we sort of develop better treatments, right? VT is a particularly severe condition. When it's untreated, it has a 50% mortality rate. The group at UCLA has taken a sort of neurosurgical approach using what is called a cardiac sympathetic denervation. You could also think of it as a partial bilateral sympathectomy, where they're cutting the sympathetic nerve fibers from T1 to T4 and part of the stellate ganglion in an effort to actually reduce some of the excitatory input that the brain has on the heart. They showed that it has an improvement in survival rates. We've done some studies showing that there's a bit of an effect in reducing anxiety, but no effect in reducing depression severity or PTSD symptom severity. So a lot of my work at UCLA has involved developing a care model for these patients. But really, how can we improve upon a treatment model, particularly given that whacking out or rotor-rooting somebody's sympathetic nervous system is a rather severe intervention and probably not likely to work for everybody. So this is the second sort of take-home question that I want to leave you with, or message, and that is really, can reducing exteroceptive stimulation help to reestablish affective homeostasis in an increasingly anxiogenic world? So a lot of the examples I gave you relate to sort of traumatic events that people might have in the world. We live sort of increasingly now, however, in both a physical world and a digital world. And the digital world can impact our nervous systems in ways just the same as the physical world. Certainly in the era of social media, and also just sort of the amount of time that we spend online. I think Zoom fatigue was something that was described in the pandemic. And I think what it has sort of made clear is that in today's modern world, silence is an increasingly endangered species. So it's encroached upon by the advancing fronts of many, mostly digital environments. But it wasn't always that way. Hunter-gatherer societies, when they've been studied anthropologically, showed that actually when quantifying the amount of time that these individuals spent, one of the most common, sort of consistently top three finding was that they would just sort of do nothing. And this is outside of the hours of sleeping. We know that sort of being still and silent is far from being inactive. It probably requires a certain mindset. And with that in mind, what I want to introduce to you is to something called reduced environmental stimulation therapy, or REST, via flotation. So this is an example of a flotation rest environment. Kind of strange, but I'll walk you through it. So it's a 8-foot diameter pool with 11 inches of water in it. The air temperature and the water temperature are heated to about the surface of the skin, so about 94 degrees Fahrenheit. The room is soundproof. And when people are in this environment, generally they turn the lights off. So there's no sound coming in from the external world. And it's so dark that not even a single photon of light is hitting the retina. So whether your eyes are open or closed, you can't really tell. Now, why do they call it flotation rest? So all of those are sort of part of the rest environment, but the flotation part, as you can see, this person is floating effortlessly on the surface. There's about 2,000 pounds of Epsom salt dissolved in the water, so more salt than the Dead Sea. It's a magnesium sulfate, so it's a kind of silky feeling bath salt that doesn't make your skin prune. When you stand in the water, your feet hit the surface of the floor, but when you lay down, your body surface area increases and you float effortlessly on the surface. This is the brainchild of my colleague, Dr. Justin Feinstein, who, after experiencing one of these environments, thought that this could be helpful as a potential anxiolytic and mood altering intervention. This is not a new concept or technology, and the older term you may recognize from this is something called sensory deprivation, but the newer terminology is flotation rest. It was originally invented by a psychiatrist by the name of John Lilly and Jay Shirley. They created this while they were at the NIMH. Initially, it was looked like this, which is kind of a scary environment. You've got this vertical float tank and a helmet so somebody could breathe. They were interested during the space race of trying to simulate outer space environments where there was no gravity, no sound, there might be no light in certain contexts. And so how would humans tolerate it? And it turned out that although there was some apprehension about putting this scary mask on, when people came out, a number of them reported feeling relaxed and refreshed. So much so that a commercial float tank was developed, which is depicted here. You could say looks kind of like a coffin. And if you have claustrophobia, there's no way you're going to get yourself or an anxious individual in there, hence the open design that I showed you earlier. But there was actually the first commercial float center opened in the 70s in Beverly Hills. Where else? It was caricatured in the movie Altered States at the end. There was a little bit of research. So here's somebody having their blood pressure measured while they're lying in a float tank. But there was kind of a stall in progress until the early 2000s. There was a meta-analysis related to stress. And then here you can see the float tank that Justin built at LIBOR when we began our research in earnest and published the first papers in 2018. This was, by the way, happening on a wave of increasing popularity. So there's a lot of float centers that started opening up in the early 2010s. Here you can see the amount of float centers in the US. And people were paying money to do this, upwards of 60 to 90 minutes, kind of similarly to the way they would with a massage. Now, what's going on here? If you think about the brain, well, if you're in an environment with attenuated visual input, auditory input, proprioceptive input, you're not moving. So as long as you're not moving, there's not much proprioception coming into the brain. You're not really touching anything when you're floating in the middle of the water. You're certainly not generating action commands or speech. So the early idea was, well, there's, maybe there's a lot of interoception happening. And this was sort of based on neurologic sort of hypothesizing as well as subjective experience. We've done a lot of studies using this particular technology. I'm not going to go through all of them, just want to highlight a few. So this was this was a single float crossover study where we had individuals float in the pool or watch a relaxing set of videos, the BBC's Earth. What you can see is that the float pool had a heightened effect on increasing serenity and relaxation. These are the same individuals. Remember, this is a crossover. So increased relaxation, reduced muscle tension, reduced anxiety on the STAI, reduced diastolic blood pressure. This certainly is a clinically significant change, but also kind of funnily an increase in cardiac and respiratory sensation intensity in the absence of a change in stomach or digestive sensation. These are, by the way, in anxious individuals who were recruited specifically for having heightened anxiety sensitivity on that ASI index. This is was early feasibility showing that we could actually get people to try a single float, and that while they felt their heartbeat and breathing sensations more intensely, they didn't do so in a anxiogenic manner. And one of the sort of theories that Justin had about why you would have this paradoxical anxiolytic response came from Joseph Wolpe and this notion of reciprocal inhibition, or sort of the idea that it's physiologically implausible for the nervous system to be in a state of anxious arousal and a state of relaxation at the same time. I'm going to wrap up. I'm just going to show you results from some feasibility work that we've done. We then took anxious individuals with high anxiety sensitivity, who, by the way, had a mix of PTSD, panic disorder, depression, and social anxiety, generalized anxiety. We randomized them to either float in a pool for 6 floats or in an active comparator involving this chair. So the light wasn't completely dark. They're laying clothed, but this is sort of to control for some of the non-specific effects. We also had another group where we said, okay, we're going to prescribe floating for one group for only an hour and in a very rigid way, but clinically, most of the time people would want to be flexible, pick to coming in as often as they want, and maybe go up to 2 hours if they want. So we just published the results of this in PLOS One. This is one image showing some of the effects. This is an average across the 6 floats. The top half is the negative effects. This scale went, by the way, all the way to the extremely negative and extremely positive end. So you can see that sort of very little in the way of negative effects, some itchiness. Some people did have some fear or panic, but they were able to overcome it. Again, it's very small. And then when you look at the positive effects, you can see that the pool environment was significantly rated more as increasing these particular, everything you see in an asterisk here. So detachment, increased energy, focus, feeling of flow, appreciation of life, pain-free existence, etc. You can see hallucinations. This is actually probably more like visual or auditory sensations, sort of like light swirls. And I have some ideas about that if you have questions. It's not sort of frank hallucinations that you think about in the context of schizophrenia. We also showed that using experience sampling, that the reductions in anxiety and depression and increases in serenity actually lasted upwards of 1 to 2 days. And that with repeated floating, we even had some evidence for baseline reduction in anxiety. Your average float study in the 80s and 90s would have gone upwards of 12 to 18 float sessions. So we don't really know whether this would continue to increase or not. So one general question I have is whether this particular approach would be feasible, not just in this anxious population that is psychiatric, but in other medical populations that have high degrees of anxiety. For example, patients with ventricular tachycardia related anxiety and PTSD, sort of cardiac arrhythmia patients. We have done some safety and efficacy work in anorexia nervosa. So this was a study that we published last year, which I won't go into, other than saying that we showed a sustained effect in body image disturbance. And my former postdoc, Emily Choquette, actually has a K Award focused on evaluating the integration of psychotherapy with float therapy to try to boost this effect. So I will, and that's something that we're kind of calling float-assisted psychotherapy. So in the last 30 seconds, I will say if you're interested in learning more about this, there's a nice podcast on NPR called Body Electric that goes into this effect of technology, and episode 5 focuses on floating. Here are a bunch of books. If you don't get a screenshot of this, I'm happy to email it to you. These are a number of books, including this most recent one that came out on interoception, that are relevant. If you want to learn more, there's an upcoming course on interoception that's applications for the course are open now. This is open to postdocs as well as faculty, both junior and senior. It's in Venice, Italy. This is going to be the second course that we had. Quite a lot of fun. So if I want to leave you with anything, it's that there's sort of interoceptive dysfunction in a lot of psychiatric disorders. There's novel approaches on the horizon, and that using these homeostatic perturbations is a useful avenue, both interoceptive and exteroceptive attenuation. But we're really at the start of this, of understanding this process. So I'd like to thank you for your attention, and I'll take questions.